Hi everyone, this is Kuhelika Mukherjee, and today we are going to discuss a very interesting topic of class 11th biology, that is cell cycle and cell division. So what is cell cycle? Cell cycle is the duration in which a new cells form, they grow and ultimately they divide. There are certain enzymes that are responsible for the cell cycle to proceed, and they are responsible for the cell division to occur. Those enzymes are called as cyclins and kinases. The cell duration cycle in each organism is different. Like in human cell, it is of 24 hours, so an entire one day is required for human cells to divide. Similarly, in E. coli, it is 20 minutes, and in yeast, it is 90 minutes. The cell cycle is divided into two phases, that is interphase, that is the preparatory phase, and the multiplication phase or the M phase. So in the interphase, it is a preparatory phase. So all the preparations that are required for the cells to divide, that takes place in the interphase. So if we see the duration of the interphase and the duration of the M phase, the interphase contribute to about 95% of the cell cycle duration, and if you calculate this, it will come to be 22 hours and 40 minutes. Similarly, the duration of the M phase is 5% of the entire cell cycle duration. It comes to be 1 hour and 20 minutes. If you see the duration of each event, the G1 phase, it takes the maximum duration of the cell cycle, followed by the S phase, G2 phase, prophase, telophase, metaphase, and the minimum duration is taken by the anaphase. So now if we discuss the interphase or the preparatory phase, it is divided into G1, S and G2 phase. In the G1 phase, it is called as the growth 1 phase or gap 1 phase. It basically decides whether the cell cycle should proceed or it should exit the cell cycle through the G0 phase. We will also discuss about the G0 phase. So here it is decided whether the cell should proceed or it should exit the cell cycle. So all the preparations of the cell cycle are done here, that is synthesis of mRNA, tRNA, rRNA, ATP, proteins, enzymes, all the preparations are done in the G1 phase. The cell only grows, there is no division of the cell cycle. So if we can consider the DNA content to be 2C. Now if we move ahead, if you see this is 2C, that is a chromosome consists of a chromatid. Now moving ahead, this is the S phase. The G1 is followed by the S phase, which is a synthesis phase. In the S phase, the DNA replication occurs, and it is the most metabolically the most active phase of the cell cycle. Why? Because the DNA replication occurs here. The histone proteins form here. As you might have studied in genetics, that DNA, they interact with the histone proteins and the packaging of the DNA occurs. Similarly, because here the histone proteins they form, there is a packaging of the DNA occurs, and as the DNA replication has taken place, now there is replication of the 2C content now becomes the 4C content. Now the DNA content becomes 4C. Centrioles also replicate in the S phase. What are centrioles? They are responsible for the formation of the spindle fibers. We will see ahead in the following processes. After the S phase comes the G2 phase. The G2 phase or the growth 2 phase. Basically, after the replication has taken place, there might be some errors that might have taken place in the replication process. So in the G2 phase, basically it checks the working and the functioning of the S phase, that if any error has taken place in the replication, it will repair those errors. And then we discussed in the G1, after G1, if the cell cycle the cell is not willing to divide, it can enter the G0 phase, and through the G0 phase it can exit the cell cycle. So G0 is basically a resting phase or the quiescent stage. So cell here is metabolically active, but it drops most of its function, that the cell is neither preparing anything for division and nor is it willing to divide. So the cell exits the cell cycle. Moving ahead, we saw mitosis. We will see mitosis. It is an equational division. It is divided into 4 phases: prophase, metaphase, anaphase, and telophase. In the prophase, the cell organelles and the nuclear membrane, they start to disappear. In the late prophase, they have almost disappeared. Now the nucleolus also disappears. There is chromatin condensation. The chromatin, it gets condensed to form the chromosomes, and in the late prophase you can see that the two chromatids, they are attached to each other through the centromere. Here in the center there is a centromere, and the two chromatids they are attached to the to each other through the centromere. The DNA content, as DNA replication has already occurred in this S phase, then now the DNA content is 4C. And the late prophase also the spindle fiber formation takes place. Now in metaphase, the homologous chromosomes, though the homologous chromosomes are the chromosomes having the same centromeric position, and they have some similarities with each other. The homologous pair of chromosome, they start moving towards the center of the cell, center of the nucleus. Okay, they align themselves in the metaphasic plate. This is the metaphasic plate, or the equatorial, also called as the equatorial plate. The centrioles, they lie towards the opposite poles, and they form the spindle fibers, as I already told you. And these spindle fibers, they attach to the kinetochores present in the centromeric position of each chromosome, and the spindle fibers attach to the kinetochores. In anaphase, what happens? The size of the now the spindle fibers, they get shortened. There is the splitting of the centromere takes place, and now the chromatids, they move towards the opposite poles. The DNA, as you can see, they have moved. The chromatids have separated. Now the DNA, the content of the DNA gives half. That is, it was 4C here. Now it becomes 2C. In telophase is basically the reversal of what happened in prophase. In prophase, the cell organelles and all the nuclear membrane, they disappeared. In telophase, the nuclear membrane and the cell organelles, they again reappear. The chromosome, they lose their identity, and the cytoplasm now contains 2 nuclei. The telophase is followed by cytokinesis. Cytokinesis is basically the cytoplasmic division. In plant cells, the cytokinesis takes place by cell plate formation. That is, there is a centripetal cell cytoplasmic division. And in case of animal cell, there is a cell furrow formation, and it is a centrifugal division. In meiosis, now we will, in this way, the mitosis has taken place. The cell has divided in case of mitosis, and now we will discuss meiosis, which is a reduction division. So meiosis is also, it takes place in 2 phases, that is meiosis 1 and meiosis 2. First we will discuss meiosis 1, which is again divided into prophase 1, metaphase 1, anaphase 1 and telophase 1. If we see prophase 1, it is further divided into leptotene, zygotene, pakitene, diplotene and digynesis. In leptotene, the cell organelles and the nuclear membrane, they start to disappear, just as it happened in the prophase of mitosis. Similarly, the cell organelles and the nuclear membrane, they start to disappear, and the chromosomal material, they condense to form the chromosome. The nucleolus, it persists, and the DNA content is 4C. In zygotene, the cell organelles and the nuclear membrane, it completely disappears, and the 2 homologous chromosomes, they start to pair themselves with the help of a synaptonomal complex that forms between these 2 homologous chromosomes, and this process is called as synapsis, and this leads to the formation of a bivalent or a tetrad. This is a dyad, and the 2 dyad, they make up the bivalent or a tetrad. Moving ahead, we see after the zygotene there is pakitene. In pakitene, crossing over between 2 non-sister chromatid takes place. That is, the crossing over between these 2 chromosomes, between these 2 homologous chromosomes, there is a crossing over takes place, and due to crossing over, there is a formation of a recombination nodule. The formation of recombination nodule, there is an enzyme, it releases an enzyme called as recombinase enzyme. And due to this recombinase, the recombination occurs. What is recombination? Recombination is the exchange of genetic material. So there is an exchange of genetic material between these 2 chromatids. Now the synaptonomal complex, it persists. The formation of synaptonomal complex that happened in the zygotene, it persists in the pakitene, and the nucleolus also persists. After the pakitene, we come to diplotene. In diplotene, the recombining chromatids, the chromatids that recombine, they start to separate from each other. The synaptonomal complex that was present here, it starts to dissolve, and so these 2 homologous chromosomes, they separate out from each other. Only the chromatid that was getting recombining, they stay here, and they form an X-shaped structure, which is called as chiasmata. Now dikinesis takes place, in which the chiasmata also disappears, and the chromosome that were the recombination, you can see that the chromosomes here, the homologous chromosomes have recombined. And then this was prophase 1. Now metaphase 1, it was just like metaphase of mitosis. They will align at the equatorial place or the metaphasic plate, and in anaphase 1 only the homologous chromosomes they will separate out. There is no splitting of the centromere. The only homologous chromosome, they move towards the poles. In telophase, now the two nucleus are formed. They are present inside a cell, and each cell they will each nucleus, it consists of tetrad of cells. In cytokinesis, the cytokinesis that takes place between two meiosis phases, that is meiosis 1 and meiosis 2, it is called as interphase. So interkinesis. So interkinesis is also a type of cytokinesis where the cell division takes place. So the two nucleus that are formed, the cell division will occur and they will go to two different cells. This is followed by meiosis 2, which is the equational division. Here just whatever happened in mitosis, the prophase of mitosis, metaphase of mitosis, this will take place also in the meiosis 2, that is prophase 2. The chromatin condensation or chromosome formation will take place. The spindle fiber formation takes place. In the metaphase 2, the homologous chromosome, they will align together. They will align at the metaphasic plate, and in the anaphase now the chromatids they separate. There is splitting of the centromere, and the chromatids they will separate, and the DNA content now becomes 2C. The DNA content was 4C here, now it will become 2C because there is a separation of the chromatids. And I have shown here taking one cell, that this cell is undergoing prophase, metaphase and telophase. Similarly, the second cell will also undergo prophase, metaphase, anaphase, and it will both of the cells now they will lead to formation of 4 nucleus, and each nucleus consisting of dyad of cells. And in this way now this entire cell will undergo cytokinesis and formation of 4 cells. Now there is, you can see that in meiosis there is reduction of the chromosome material. So in this way, after cytokinesis, 4 cell formation takes place and the meiosis completes. Thank you.